Thank you, Nico. It's a real pleasure to be here. Really honored and pretty much impressed by the amount of Pichia experts in front of me. So I hope I can charm you by explaining an update, giving you an update on our nanobody platform and our Pichia platform and function of nanobody production. So I will talk about the platform, how it looks like, and secondly I will talk about strain development, and eventually I'll have a slide or two about what everybody's talking about these days, how we will implement artificial intelligence into our platform. So first introduction on the company itself. So as Nico mentioned, you may know us better under the name Ablings. So we were the first biotech company who had the ambition to develop nanobodies into biopharmaceuticals. That was in 2001. And it takes a long time before you get something to the market. So in 2018, we finally got there. It's like a chance in a lifetime. And then of course you get a lot of attention of the big pharma. And in 2018, around the period that we got this commercialized, we were acquired by the big French company Sanofi. And since then, we're more known to the site, Sanofi Ghent, where we are located. And if we have any Pichia issues, we just cross the street towards Nico Kallewaard's lab. So nowadays, we are having, so the first, maybe I should mention that, the first nanobody is raised against a target to help patients with a very rare disease called thrombotic thrombocytopenic purpura. And we are now having 440 people working in our labs here, not even including our favorite employees, the llamas. And most of them are scientists focusing on nanobody development and bringing new medicines to the market. And the data I will share will come from the host and tool creation group and from the upstream processing team. Next slide. Yeah, but of course, for the ones, the people that are not familiar with nanobodies, 2 slides to explain that, because it's a fantastic tool and because it's so simple actually. So the story starts with this professor here, Professor Hamers, who in 1989 teaches his students about the structure of conventional antibodies. And it had some hands-on experiments involved. And in 1989, the students mentioned to Professor Hamers that their findings did not match the teaching. And that was always a question of coincidence, because that was because someone in his lab was doing studied infectious diseases with camels. And in 1989, for the first time, he gave blood of a camel to the students instead of the regular blood of a rabbit. So they were all witnessing the discovery of single domain antibodies in camels next to the conventional antibodies that Camelidae family have. So the work of Professor Hamers is important because he then pinpointed the minimal domain of a heavy chain only antibody that could still bind its target, and this is only like 350 base pairs long. In the next slide, you see how fantastic this is. So just because nanobodies are coded by a single gene, you can take a nanobody against target purple and fuse that to another nanobody against target green, and fuse it to another nanobody against target yellow, and the only limitation is your imagination. So you still get a single gene coding for a single protein with multi-specificity. So the fantastic thing is that you have here purple, yellow, green, great potency, but if you want to explore, if you can increase the potency, you can play around with the linker length between the two nanobodies. You could use a linker such as glycine-serine combinations of 9 amino acids, or you can extend it to 35 GS linkers. So you can play around there. What you also can do is you can switch the order of these building blocks. It can also affect potency. You can also multiply multiple of these nanobodies, and also as such increase the potency. But the big thing is that you also change the biophysical characteristics of these molecules. And that is important because you have to have a filter to select that nanobody combination that falls under the denominator of medicine. So this combination could have a solubility of 1 milligram per ml before it precipitates. You just switch the order of the building block and you can have a solubility of 100 milligram per ml. And that makes a big difference. So you soon find out that you have thousands of possibilities to evaluate. So you need good filters, and of course we are interested in which of the combination expresses the most, gives you double digit in Pichia. Next slide. So this is a bit of the history of our expression platform. In the beginning, in 2001, we made the classical mistake that all biotech make. We use E. coli for the expression. And you can see we were able to have one process developed in E. coli. It's so intensive. And here I give you an example. E. coli expressed our nanobody here, and the same nanobody expressed in Pichia. Yeah, DSP is having a ball. That was in 2007. We said it's worthwhile paying RCT. So we switched to the X33 strain, and you can see we were in a minimum of time we could have 4 process developments and 4 GMP runs, and went to the clinic with those 4 nanobodies. Then later on, we used our own strain. I call it the Neri strain, otherwise I break my tongue. And we engineered it, and you see that engineering this strain, we had 11 GMP manufacturing. So we done 11 process developments, so it really went fluently using Pichia in the production of nanobodies. And then when we were acquired by Sanofi, the message came from the big pharma, you should comply to the health and safety regulations. That methanol, you should kick it out. Now that was like, that was a crisis, because we were so used to work with methanol that performed so well. I had nights I was laying awake at night, of course. So we had, also for eco-environment, there was an argument to shift to a methanol-free system. And so I will talk about later how we manage that. So we now have one process under methanol-free conditions under GMP manufacturing in Frankfurt with the colleagues in Frankfurt. And in the next slide, if you want to start up a nanobody company, you need an expression platform. It's always good to know what it should perform, what it should deliver. And if you have a program, you go through several stages where you need several amounts of material. So in the first stage, it's of course the research stage. You don't need much, but as I said, you sometimes have hundreds of different nanobodies you want to evaluate, and that can still be around 10 to 500 milligram production that you need. So later on, once you have your handful of preclinical candidates, you do developability, which of the 5 should go to the clinic. So there you find that you will need 5 to 20 gram of material depending on the drug substance concentration that you will have for that particular program. Immuno-oncology programs, for example, are typically lower concentrated, and therefore less material is needed than for other programs that have sometimes 100 milligram per ml concentrations. Once you know which is your clinical candidate, then of course you do process development. So also analytical methods need to be developed. You study the formulation of that particular nanobody. So you find that you need to produce 100 gram, for example. And then of course, the large-scale production in function of tox material that need to be delivered. So you need the capacity to deliver up to 1 kilo of material of the nanobody. Okay, next slide. And these are our workhorses to do that. So these are the numbers of 2023. So over 1,000 fed batch fermentations are being done in our labs. And you can see here the ponies, the amber fermenters, the ones that deliver us 10 to 100 milligram of material. They are 700 runs per year. Most of the runs are done with the amber. We also use that for design of experiments, for example. And then we have the robust vessels, the 2 liter to 5 liter vessels. We do over 300 runs with these. If in vivo studies are required, then we need sometimes more than multiple gram levels after DSP, of course. And then once we do the tox material, and then we have the steel vessels. You see we have 30 liter, 100 liter, and a 400 liter fermenter is the max. We also have this special one, the single-use fermenter, 300 liter from Thermo Fisher. And that is, of course, for the high-potent nanobodies. If you have highly potent nanobodies, you produce them in steel vessels, especially in the GMP manufacturing setup. You need a cleaning validation. And using single-use fermenters, you just throw away the bag and you don't have that issue of cleaning validation. That's why we use the single-use fermenter for Pichia. The process is a little bit different than in steel. You have to watch out for oxygen uptake, etc. But it works perfectly. Next slide. And this is how it looks like, our lab. You have here the 300 liter SUF, you have the 100 liter fermenter, and there the 400 liter fermenter. And then the clarification. So we have some mixing containers, and you see here the continuous centrifuge. And regarding clarification, in the next slide, you see that we typically use for the smaller fermenters, like 2 liter up to 2 to 5 liters, we use the batch centrifugation for clarifying the cell broth. I forgot to mention, the nanobodies are being secreted with the alpha mating factor, so we don't need the cells. And then for the larger scale, we use the continuous centrifugation in combination with the depth filtration train. For the highly potent nanobodies, we use the hollow fiber clarification step. And of course, you need a team to do the productions day in, day out. So it's important that we do not burn our employees. So we agreed for the Ambers, for example, that around 16 runs are being done per week per operator. Especially when you do a design of experiments, you have mixed combinations and that takes more time than a generic production, of course. For the 2 to 5 liter runs, we have 4 runs per operator per week. We had in the past 10 liter runs as well, but that's too heavy for the employees to carry. That's 40 liters that they sometimes have to carry, so we had to omit that from a health and safety perspective as well, since we are Sanofi, so slavery has stopped. And then for the larger scale, of course, 30 liters and steel, we use 2 operators because you have preparation time before and cleaning afterwards as well. Very important to motivate your employees is that in a program, they start doing the design of experiments, the process development, and they grow along the program up to the large-scale fermentation. And as such, all employees are able to work with small scale up to large scale, which gives us enormous flexibility. If someone goes on vacation, everybody knows how to work with these things. Okay, next slide. And now, what you're all waiting for, especially the people from Texas, how I had a bet yesterday on the boat about what the titer, the average titer will be of the nanobodies in Pichia. Well, here it is. During COVID, we had a lot of time and we cleaned up our data. So we selected all the different nanobodies that we have expressed in Pichia under generic conditions. And these are the numbers that we came up with. So the titers vary from practical 0 up to 20 gram per liter. Yeah? And it's in the methanol-based system. We have an average titer of 3 gram per liter. So I get a beer this evening. Yeah? And this is about almost 850 different nanobodies expressed. But the surprising thing was, you would expect that lower titers was expected of the more complex formats of the nanobodies. And that appears not to be the case. If you here see the monovalent nanobody fused up to a hexavalent format, and if you look at it, there's no trend in a dropping titer with increasing formats. And I'll come back to that later. And now, the point, how does the methanol-free is performing here? Because, well, look, The numbers are smaller. We had 156 different nanobodies, but the titers are slightly higher. So that's my second beer I deserve, right? So 3.7 gram per liter. We were very happy to have this in-house. And again, same story. With increasing format, we did not see a drop in titer. So this is like the individual building blocks. I explained that the individual building blocks, probably the folding kinetics are fast enough so that you don't get real spaghetti into the ER and the Golgi network. So Pichia and nanobodies, it's a perfect match. In the next slide, yeah, this is an overlay of methanol-free versus methanol-based, and you see they perform equally as well, at least. So if we want to go fast to humans, what is very important that you have a very close interaction between our discovery department and our early CMC department. And that is actually what we do. We want to have a first-time right approach that early on we select the nanobody that is expressible in Pichia with the right quality, and then later on we don't have to switch, for example, to a different strain. It saves us resources. So that interaction between CMC, host creation team is very important. And we early on in the methanol-based system, many years ago, we started engineering our strains to get the right strain available. That's on the next slide. And what we've done, we went to Pichia conferences, we scraped the internet, what you have been publishing of auxiliary proteins that could increase the titer. And we selected 22 chaperones and evaluated them for improving the expression of a very bad expressing nanobody here, a bivalent nanobody that expressed in the wild type Pichia at 0.1 gram per liter. And four of these auxiliary proteins that we evaluated seemed to improve the titer significantly. RPPO was a ribosome-associated protein, CAR2 HSP family member, and then of course the big winners were HAC1 and PDI. So what we've done was, on the next slide, we combined those four auxiliary proteins, put it in the Pichia strain under the control of the AOX promoter, the same one as we use for the expression of the nanobodies, and we did some feasibility studies with other nanobodies. Here you see a trivalent nanobody, wild type, that expressed at 1.8 gram per liter. That was not bad, but when we expressed it in the strain that co-expressed those four auxiliary proteins, we jumped to 2.4 gram per liter, and here a tetravalent nanobody in the wild type, we had 0.3 gram per liter, and it nearly jumped to 5 gram per liter. So we were very happy with our new platform strain. We used that for years. And in the next slide, here you see the average titer in the wild type for 54 different nanobodies was around 1.4 gram per liter. And using that platform strain, we could double that titer. So yeah, indeed, so this is again the same story, using that platform strain, expressing those four auxiliary proteins in the methanol-based system. We did not see a drop in expression level with increasing formatting, but we had the impression that in the wild-type strain, that could be the case. That we did see that the wild-type Pichia cannot handle these kinds of complex molecules. Okay, and then the story about the methanol-free, we had to change. So we evaluated several types of methanol-free expression systems, and my colleagues in Frankfurt, Daniel is here as well, he identified the two promoters that were worthwhile evaluating, the glycerol regulated promoters, PDC, from catalase, and the formate dehydrogenase from Hanseneur polymorpha, the PDF promoter. So using these two promoters, we developed a new platform strain in the early strain that co-expressed HAC1, because you've seen that had a strong effect on the expression. And the HAC1, we put it on the PDC promoter, and the nanobody under the PDF promoter. Seems to be a good combination. Next to that, we also had to look into the workflow, maybe one back. the workflow of a methanol-free production, is that still within working hours? So that's what I'm illustrating here, and that seems to be the case. Our employees did not bother too much, so we start pre-culture on Wednesday. Thursday the batch is starting, so the fermenter are inoculated. On Friday we start the fed batch, and then automatically on Saturday, the glycerol feed is adjusted and the induction is initiated over the weekend, and harvest can be done on Tuesday. So pretty much similar to the methanol-based system. In the next slide, we had a feasibility study using that methanol-free system. We expressed 5 nanobodies that we knew very well regarding quality and titer in the methanol-based system. And in the blue bars, you see the expression of these 5 nanobodies using the methanol-based system, and that's around 2 to 3 gram per liter cell broth. And then the methanol-free system is in the orange and the gray bar, where the gray bar we co-expressed the HAC1 protein. And you immediately see that the effect of HAC1 is very clear. It jumps up the titers every time HAC is present. Regarding the comparison between the methanol-free system, you see that two of these nanobodies do not match the levels that we reach with the methanol-based system, but the others, it's in the same ballpark. So we're pretty happy with that. It doesn't mean, the message is, it doesn't mean that you have nanobodies that are expressed in methanol-based, that they will automatically also have a good expression in a methanol-free system. So you need to screen for those nanobodies that are fit the platform. The next slide. So this is an overview, methanol-free system using the auxiliary protein, 3.7 gram per liter for 156 different nanobodies, and no effect of the valency on the titer can be observed. Okay, next slide. Okay, so as I explained, the number of combinations you can make with nanobody is just enormous. You need kind of a good filter if you want to find out which of these combinations have good expression in a fed batch fermentation. And that is sometimes problematic because you probably all use 90 cell well expression to screen clones, and then you hope that it translates to a fed batch fermentation. We were thinking, well, this is not always a one-to-one comparison. You sometimes see surprises. So can we turn to artificial intelligence to have some predictivity? So in the next slide, I explain you how we typically do our screening regarding expressibility in 96 well plates. So here you see a construct, a trivalent construct, with 3 nanobodies connected, yellow, green, blue, and we vary the linker lengths in between. These are over here. And here, the same construct, but we switch the 2 building blocks. And we also evaluate the different linker lengths. We transform all these constructs to Pichia with pick clones, and we express them in 96 wells. 20 clones are being picked, and the expression in the 96 well plate is being evaluated using bilayer interferometry using protein A tips. And that is the ordinate signal that we get. So you would expect that these constructs with a green nanobody in front of it are the best expressors. Same story here, we have a tetravalent nanobody. We switch order of the building blocks over all these constructs. We also evaluated the 35 GS linkers, the 9 GS linker and the 20 GS linkers in between the nanobodies. And from these results you would expect these are the best expressors with a favorite for the 20 GS linker between the nanobody building blocks. If we put some of these clones into fermentation, that's on the next slide, you see that 35 GS linkers gives us 3 gram per liter in fed batch fermentation. And indeed, the smaller linkers between the nanobody blocks gives a higher titer, 14 gram and 7.9 gram. But see, the resolution between these two cannot be reached in a 96 well expression. So please do a lot of research to make this better, because That is a pity that we miss these clones sometimes. And therefore, can machine learning help us? So based on, I think on the next slide, the question was, what shall we use for the machine learning? We had thousands of titers of different nanobodies. Should we incorporate the coding sequence or the the primary sequence? And we believe it's the primary sequence that is mostly important, because if you make a nanobody, you need to humanize. You need sometimes change some amino acids, not to have some immunogenicity. And we often find that if you touch the wrong amino acids, you get energetic clashes into the nanobody, and that affects the folding kinetics of the nanobody in the cell, resulting in a lower titer. One amino acid mutation, you drop the titer from 1.5 gram to 0.4 gram, so you have to watch out for that. So we think that relating titer versus amino acid sequence, you could make a model. That's what we've done, and bear with me here for a minute on the next slides, because that's work from, you can skip that one. That's work from the bioinformaticians. We gave 1,000 sequences, and because we had more data of the methanol base system, we gave 1,000 correlating with 1,000 titers. And the biomathematicians, they start working with a language model, and we said, look, can you make a model that predicts different nanobodies, select different nanobodies, or put the nanobodies into the basket of low expressing versus the higher expressing titer. And on the next slide, this is the result. So we gave them 100 new nanobodies, of which we knew what the titer was, and we asked the model, how do you classify these 100 nanobodies? And he put around 50 nanobodies here under the low expressing type of nanobodies, and then the machine, that's the box Richter plots, he says, look, and these nanobodies are the better expressing nanobodies. And then we compared our results of the fermentation, and these are the black dots. And you clearly see that the machine is right, that the black dots really, from what the machine predicted, that were the lower expressing nanobodies, you see a significant difference that he really could identify the better expressing nanobodies. That might be the future, where we give the machine thousands of combinations of nanobodies that probably do not differ much in potency, but make a big difference in manufacturability and the cost of goods, and of course, that's important. Okay, and then my next slide, I have to thank the team at Ghent, the USP and the host creation team, and of course our colleagues in Frankfurt, who also have a big expertise in Pichia. And if you would, on the next slide, would be interested to join Sanofi and working on Pichia, we have several sites where they have Pichia expertise. Toronto is there. We have, of course, a concentration of Pichia experts here in Belgium, in Ghent, Frankfurt next door, and south of France is also a very nice place to study Pichia. And even more south here in Buenos Aires, where we make a vaccine in Pichia as well. Thank you for your attention.
A very nice talk. I was interested in this AI prediction, so that was just based on the sequence comparisons of each of those domains. How was it done exactly?
Yeah, so we had the sequence of the nanobody, and the full sequence was put into a language model correlating to the titer. But bear with me, I don't know the details.
Okay, but I want to take that one step further. Now, obviously, if you know the antigen and the antibody structure, now you can use AI to predict also binding abilities along with production abilities. Have you tried that?
That is something that we are working on, and it's not an easy task, but eventually, I'm sure it's going to happen. Yeah.
Thank you, fascinating journey. I have two questions. One question is regarding post-translational modification and immunogenicity. So Like, it's a strong objections. What's your experience with FDA and registration for that? And the second one is, when you're introducing so multiple different alterations in the Pichia strain, how stable is that? Thank you.
Two perfect questions, yes. So the first question, FDA is well aware of the modification that we have on nanobodies. It's certainly no N-glycosylation on top of that, so that is engineered out. So what we have is sometimes like a percentage of O-glycosylation can be found on serine and threonines. And no big issues. We have no data that triggers a kind of immunological response with the patients. So that's okay. It's not even a criteria, the percentage of O-glycosylation on nanobodies. And the second question, indeed, I'm surprised that that we don't talk more about the instability of clones, and that is in the flow of our strain engineering process. We look at the stability of our clones, and you can mimic cell banks and evaluate whether you see a drop in titer. And that often happens if you do random integration. You hit something that results in a clone that may express very well, but appears to be unstable, and you lose copy numbers. And that's the reality. So we screen for that. Yes. Yeah.
And it's very impressive work, because before this conference, I'm very skeptical about like methanol-free system. But after talking to so many people, and that totally like turned my mind and think that this is very promising system with all these powerful promoters. But now my question is, we also have the same challenge when we want to put different vaccine unit into one fusion protein and express it as a whole product, and so it can broaden the protection of a certain vaccine. But we have this challenge of when we put, if we express them separately, they express well, but if we fuse them with linkers, and we are not getting things expressed. So I'm very impressed to see you're able to express like molecules like up to like 90 kilodaltons with the Pichia. For us, something usually if over like 30 kilodalton, then it just won't express very well. So
yeah, well, this evening with Bear, I give you free consultancy, but it's a trial and error. I mean, we're so spoiled with nanobodies, as it's a perfect match, and you cannot project our luck to your type of molecules and fusions you want to make. I would say it's a trial and error. I can't help you.
And if you like, what do you think of the order of the combinations? If you put an A, B, C, or like B, C, A, do you see like that we have a huge impact of the expression?
Yes.
Yes.
Yes, that can happen. Even like single monovalent antibodies, they can express all well. You fuse them together, and suddenly it's a drama. You don't get any expression, and you don't understand. And then you leave one nanobody out, and you get lucky again. So having a good filter on the train is important.
As Peter is offering free consultancy tonight, we will have just one last question.
Yeah. Hi. Thank you for the talk. I was wondering, based on all of these experiments, how much, and these very impressive titers, how much would you contribute to strain engineering versus process engineering? Like, I can imagine that there's a very big difference antibody depending on each antibody, but if you would have to put like some ballpark number on this?
Certainly strain engineering is the heart of it. And then design of experiments to optimize your process focuses less than on the titer, but more on the quality. And that is all because in the beginning, when we did fermentations, we start optimizing. Every time we found that, oh, this condition is better, and it seems to be a generic condition. So now our generic process is pretty close to the optimal process. I forgot to mention, we use complex media to prevent protease activity on our nanobodies. but that is our generic way of working. If you switch to a synthetic media, there you might need more optimization, and there you could perhaps double the titer and the quality as well. Does that answer your question?
Yeah, thank you.